one of the advantages of the local healthcare community. So this is Sydney Vital. We're a network organization. We sit within UTS, Uni Sydney. We work with Macquarie University. We've got the MARTA, where we have collaboration with, etc. The vision is that we establish a competitive network of excellence and try to conduct collaborative and with the operative word being collaborative translational research to improve patient outcomes. So it's directly related to patient outcomes. If not, then it becomes, from the viewpoint of CNSW, almost unfundable. And the mission then is to encourage and promote innovation and collaboration among preclinical and clinical researchers to support cancer research across the pipeline. It's motherhood statements, but this is what we're trying to do, and I think all TCRCs in essence are trying to do this. So you do that by enhancing collaborations, which we do with Macquarie University, I think, being a prime example, but also aims to give just a few examples at University of Sydney. A governance structure, meaning that how do you support basic research, how do you support clinical research, is trying to, if you like, facilitate the process of writing, for example, the grants that you have to go through Sydney Vital to go to CNSW to apply for grants, like research equipment grants or total translational program grants. And to develop the essential research infrastructure, I think we've been quite successful, and I'll give you a couple of examples in our research equipment grants. And certainly this year we've, again, been successful in 2 of them. And the essence is that I think if you have a translational cancer center, then that is a stepping stone, if you like, to get into the money that CNSW has stashed away, if you like. So how do you link the right people to do research? How do you link them, the clinical research with the basic research? I think at Sydney Vital, at least we've tried to incorporate as many of all people in the research pipeline, if you like, at our management team. And there's always room for improvement, of course. So how do you align? How do you identify the right questions? I think this is where, for Sydney Vital, and certainly for a local health district where there's so much need for clinical work, and thus clinical questions, and thus basic research related to clinical questions, we have to sort of identify the right questions. I think this is a process, an evolving process, and I'll come later to how we sort of think and have been thinking about this at Sydney Vital. I think we've done a number of research educational programs, certainly for our community, but also for our local clinical healthcare. We've tried to set up, for example, an educational evening for researchers solely. Hasn't yet taken place, but I think that's still on the card. So what we've done is educational series with, for example, on rectal cancer, or on lymphoma, or on lung cancer, where we had a dinner meeting with about 60 interested clinicians and some basic researchers across one topic. And we hope to be doing that in the next year for the basic researcher community. And then how you did the dissemination, of course, is, and I'll come back later to that. Some of the things that we've done that have come out of the past one or two years, and these are just a couple of examples, where we have actually gone and done something that has made an issue or difference in the community, such as, for example, the PetNet grading system, which makes the up to now really differentiated number of patients and differentiated patient population almost more you get a handle on them in terms of what sort of groups you're actually looking at. And the same for example, for the lymph node involvement in malignant colorectal polyps, where we've now come up with a tool that prevents probably 50% of the patients have unnecessary sigmoid resections, but still is a quite morbid procedure. The collaboration, of course, are links with TCRCs. This is one example. We've got a number of studies that are examples. And I think that's the future. This is what CNSW probably wants, is more collaboration instead of the original model where it was more competitive between TCRCs. And that's fine, I think. Our local health network for the GPs is interested in collaborating with us. Sydney Health Partners, I think I'm just going to show an example of one of the studies that links all of the partners at Sydney Health Partners. And of course, we've got industry partners that have funded, actually quite liberal, quite liberal in funding some of our educational series. So, research. So, what have we done? And this is certainly thanks to the work that Debbie Marsh has been doing. But we now have global informed consent, which means that one of the basic requirements, I think, for further research has now been met. There's global informed consent, which means that virtually everyone who comes through the door could be asked to contribute to research projects and to buy specimens. So we've got a biobank pilot. So you have the global informed consent, but how do you then biobank? How do you streamline that? Which is now a project that's underway. And for our colorectal cancer study, that's become instrumental because the way the specimens are collected was one of the bottlenecks, and that has been solved. And there's, of course, a very extensive surgical pathology database. These are some of the research equipment grants that we've been able to get into the campus, and one of the recent ones is, of course, the animal irradiator, which I think is probably going to be instrumental in doing some very specific local research that is not available in other TCRCs. And for this year, we've been successful in 2 REGs. This is how we distribute. So, of course, we've got the website, we've got social medias, we've got GPs events. The educational events have been very successful. We've held probably a number of 16, I think, in total now. We've got our annual conferences, and we've got a number of the symposia. So this is what we have grown into in 2014. We're now 290 members. We've got probably in Cat 1 and Cat 2 funders publications, some 300 publications, 260, so it's growing. And there's leverage funding. Now, the leverage funding is always interesting because this is CNSW leverage funding. If you look at the real money in leverage funding, it's probably a bit less, but this is what we are reporting to CNSW. So I'll skip this one. So this is what it's all about, I think. This is how do you build a community, and how do you make what you do significant for these people, and that then feeds back into what we can do. Because this, for example, the neuroendocrine charity event was a fundraiser, but it's also really great to be able to communicate to the community what you're doing and what sort of difference it makes. And this is true for all the events we've done. One of the particularly successful one was the patients and community event with neuroendocrine. I'm just saying this is the link that we're looking for in the TCRC, I think. And any cancer research is that it gets fed back to the community. That's where you actually want your results to be. Done this, we've done this. So, for example, this is the collaboration with AINST. It's been particularly successful because this has been almost like a runaway success, which started from just a visit to one of the presentations at the Nano Institute, to now a project grant application for NHMSC in how do you improve lymph node detection in solid tumors. And from that, how do you then treat lymph node metastases in solid tumors. And if you look at the collaborators there, a lot of them are, of course, from the local campus, but there's Westmead, there's University of Sydney, there's people from MD Anderson in this project, and certainly in the project grant. So it's collaborative, it's multi-TCRC and multi-university. This is the Sydney 1000 study, another example of how we think you need to have larger projects that then become, of course, a clinical research project, which ties in a number of basic research opportunities. And again, clinically, they're from this campus, so that's the clinical research. But if you look at the translational research, it's actually across multi-TCRCs. And that's, I think, the way certainly we see how things should evolve. But I think CNSW is also very much on this pathway. And of course, the Colin Tumor Bank, which I think is one of the major core facilities, if you like, that underpin the opportunity to do research, but also have a very, very large bank of data. And one of the examples, of course, is Innovate, which is led by deFazio, but it's also this large collaborative multi-TCRC, multi-hospital research endeavor. Now, what are some of the issues locally? And this is to do with the Colling Review. Colling Review was and has been done, and it's to do with how do we look at how Colling has performed, and are there any ways to improve the way the Colling is functioning. So this was the Sydney Vital letter. On the left-hand side, this letter I wrote, I sent it to all the members. Many have sent, of course, their own submissions to the Colling Review, but when I sent the letter, there's probably 10 of 250 members who actually commented on the letter, which thank you very much for commenting, but it's only 10 of the 250 members. Anyway, this is what I think is sort of a, if you like, a condensation of what has been fed to that committee. So, strength of the Colling and the local health district, well, some of them have been mentioned. State-of-the-art clinical facilities, large incidence of cancer patients, experienced research, both clinically and basic research. There's a quality undergraduate and large postgraduate education program. There's access to key opinion leaders. There's global consent across public and private, which is very important, I think. And there's a mature biobank facility and an extensive surgical pathology resource. Weaknesses, we're isolated from the University of Sydney campus, and at times that feels quite deliberate, and it's probably unilaterally deliberate. That's how it feels. Whether that's true, well, there are signs that some of it is true. We have a local health district where, although they preach one thing, I don't think their clinical and research agendas are aligned. There's a lack of integration. That's what we're lacking, is how do we integrate clinical data, which have to be real-time clinical data, with biospecimens and surgical pathology. There's a dilution of resources. We feel that maybe we're wanting to fund all research within the LHD, and we then have no strategic vision to invest or divest. There's a lack of transparency in how the funds, the philanthropic funds get distributed. There's inadequate infrastructure report for facilities, inadequate administrative support, and there's differences in systems between nodal and local health district and the university in terms of IT, HR, and financial management, which are sometimes quite obstructive to basic research and clinical research alike. So these were then some of the solutions. Again, some of them motherhood statements, but I think they're true. So how do you build synergy between university and LHD? But you have to build them on what drives them both. And I think the main issue is that they need to be based on clinical questions. That's for our local health district. There should be a strong but also budgeted commitment from university and the LHD. We should probably invest in health economics, epidemiology, and bioinformatics at the Colling. I think we need to have financial support for tenured research positions for infrastructure and facility management. There needs to be this transparent process. That's what we've talked about. We need to develop the Colling brand, but maybe we don't need to develop the Colling brand, but we should sort of have a Colling Institute and a different name for the research, for example. Of course, the community engagement, and that's why we have David Meredith, who's almost like sleeping at this campus, but in terms of the connection between the university and fundraising. And I think most of it is, I think, down to the almost below the two lines, is how do you promote interaction and collaboration between the disciplines, certainly with clinical and basic disciplines. And as Rob, as a cri de coeur, say, let's have an operational attitude and not an aspirational attitude in terms of maybe we should get things done and not talk too much about stuff. So at Sydney Vital, we think what we want to have is translation research capacity. How do you build that? By integrating clinical centers of excellence, supply specimens and surgical pathology, and I'll come back later to show you what we've developed at Sydney Vital as a starting point. Connie, could you take over and expand a bit more on transitions from basic research to the other?
Right, so I'm Connie Diakos. I'm here, been asked here by Alexander to provide a bit of a perspective from the point of view of someone who's spent time in both camps. So I started off as a basic science researcher, and I'm now a clinician medical oncologist. So I started off as a basic science researcher, and then I'm now a clinician, so I'm a medical oncologist at Royal North Shore and do some private work at the Melbourne Cancer Institute, which is a co-located cancer treatment centre, and as well try to be a translational researcher anyway, upstairs in the Bill Walsh Translational Cancer Research Lab as part of Colling here. So when I started off as a basic science researcher, I was looking at drug discovery and mechanism of action, and back at that time there was located on main campus, and there wasn't a whole lot of collaboration with clinicians at that point, but we started to develop some of those towards the end of my time there. I could see that the difficulties that all of you recognise and face, which is continued funding and employment, and so at that point I sort of had various advice and made the decision that I'd go down the path of clinical medicine with the view of returning into research at the most opportune time during that. So I guess that's sort of, I think there's been basic scientists who've seen me as a bit of a sellout by doing that, but I guess the bottom line is it's allowed me to keep bread on the table. That's kind of the basics of it. About halfway through my medical degree, I continued to postdoc and started to realise then that even just with that little bit of medical training, it certainly was enhancing my basic science research by giving me a different perspective on things, allowing me to ask some more clinically relevant questions and to approach things in a way that you could certainly see the translational benefit from that. Now as many years out of research, going through medical training, returning back 20 years ago in an area that's very different to what I was doing before. Now I'm doing research on editing in Bill Walsh, which is co-located on a maternity campus. It is significant to me as a that the argument. So I certainly see that the place of basic science research is in translational research is essential. There are questions that cannot be answered in clinical setting. It's not safe to do so.
bring it into the clinic from bench to bedside, whereas a clinician might want to see that it changes policy.
So
How do you marry these different expectations?
yeah, well, I see it not just going from bench to bedside, but also from bedside back to bench, because we do see, you might be instituting a treatment in a patient and trying to work out these days is what's the biomarker, what group of patients should we give these treatments in, what sort of responses can we expect to see. So, we can provide clear
Director at Epworth, Director of Research at Epworth in Melbourne, who's some of the issues of what actually do health providers want. Thank you.
Okay, so I'll speak into the microphone a little bit, but I'll hopefully project anyway. So I'm going to take a different tack to what you've just been speaking about, and sort of speak from the perspective of someone who's engaged with senior executive about exciting them about research across it's a private not-for-profit, I think about 1,400 beds, so it's a pretty big organisation. So it's probably about the size of an area health district as a group of private hospitals, but previously with St John's. But also from the perspective of having been on research committee of the NHMRC for 6 years and being involved in various discussions around the MRFF and a reviewer for the CINSW, which probably scares a few people in some respects. One of the documents that was very, very important, I was actually on the board of ASMR when this came out, was a review, a structural review, which ASMR at the time was saying don't do, but the government of the day decided that they would do, and they commissioned Simon McKeon to chair, was a review into the health and medical research framework in Australia to say, well, how are we doing? Where's the money going? Is it going in an appropriate direction? And this is at a federal government level. There'd been the 2 doublings of money into the medical research endowment accounts that had grown from about 200 million to 400 million and then to 800 million over a number of years. But the research sector was saying, things are terrible. I actually read an article just recently that said, it's never been worse. We've never had less money, which is garbage and annoys the hell out of government, because actually you've never had it so good in many respects by a pure dollar amount. And government will remind you of that, and they'll say, well, Treasury's appropriated more today than it ever did many years ago. So those kinds of arguments don't win you friends when you're in federal ministries and various things of that nature, because it just sounds churlish. So the idea was, from this review, to say, where should the money be going? Should there be an increment to the medical research endowment account? Where would you spend it, and what was the return on investment? And one of the problems is that government doesn't completely believe the idea that ASMR, and I was party to this, was writing that there was a 2.3 times return on investment. We're saying, well, that's kind of true, maybe not totally true, because you're really building that on CSL, and Cochlear and a couple of other instances, but you're not actually looking at all the things that don't make money. So again, not something that went down particularly well. But the review was fantastic, apart from the fact that a lot of researchers, I came to the one in Sydney when they were holding the town halls, would demand of the panel why they didn't get their research grant. So when I spoke to some of the panel members, every city they went to, someone complained about why their grant didn't get funded from the NHMRC. And one of those things when I'm talking about translational research is pitch your message appropriately. What we, in government we hear constantly, and even as a person who now has a budget that we give that away to people, is if you sound bitter about your money and you make it fairly selfish, it doesn't sound like you're actually trying to do something to improve the health of people. You're just after your own tenure and after why your grant didn't get funded. So the focus of this was very much about effective and efficient healthcare founded upon evidence created by research. And I'm going to take you through a little bit about the evolution of this document into the MRFF and what that means for you in terms of funding. I mean, these are really important statements that set the agenda for in constructing your applications to be meaningful in a translational way. Improvements in healthcare can only be achieved through research. This is about as clear as it can get, that they do believe in this, that research is important and it's integral to healthcare delivery. Now, I asked this question of many groups. What, if this was true and you were running any other organisation, how much money by proportion would you invest in R&D? What's a reasonable amount to invest in R&D in a large company that is, what, how much? 10 to 15%, yeah. So roughly, what proportion of the health budget do you think goes into legitimate health research? Forget the TTR because that's a bit of hand-waving. How much? 0.2? Yeah, that's actually about right. It is a very, very modest amount of money. It is way below what anyone would reasonably expect to put into an organisation that was dependent upon innovation, that was dependent upon research for doing its work. I mean, there are organisations, pharmaceutical companies, that spend in excess of 30% on R&D, simply because they are so research dependent. And you would imagine that healthcare should be the same. So where's this got us? I mean, this was some of the comments that were in there. I've just picked out a couple, which was enhance the capability of NHMRC and researchers to support policymakers. So how has that actually been enacted? Anyone read the budget papers and have a look at the NHMRC budget? What can you tell me about that? So anyone in this room, have you read the budget papers on funding? Okay. That in itself, you know, if you're serious about wanting to know where government is thinking, you actually need to read documents like that, particularly if you're in a senior position. So the budget, they cut the NHMRC budget once again by about 1%. So that's the actual administrative side of it. So the NHMRC is shutting down its clinical trials unit, which has been actually driving. I've worked with them very closely to deliver policy around enhancements to clinical trials. It's gone. They've lost all the staff. So they've year on year diminished the capacity administratively for the NHMRC. Now take the Medical Research Endowment Account, which funds all the grants, fellowships separately. What proportion of increment did that receive this year? Anyone hazard a guess? No? It's CPI, 1.2%. So again, it's gone up with CPI, but it's been pretty much static for the last 8 years. It's about $850 million. It'll be $870, but with CPI, that's sort of eaten up. So again, grants are going in size. You're getting more 5-year, 4-year project grants. So the success rate, as you should know, is going down at a rapid rate of knots. It was 21% when I was on research committee. When I left, and it wasn't due to me, it ended up around about the 12% mark. It'll probably pitch below 10. So these are things to be really worried about. So this embedding of researchers within government policy departments, is that happening? Not really. This is the problem that government worries about. And again, I would credit myself as being a basic scientist originally, but now I spend more of my time reading Productivity Commission and Intergenerational Report and trying to figure out what government's thinking about where we can get the money from and about how area health services might actually be excited to invest in medical research. This is the problem. And from this graph, you can actually see, I mean, don't worry about too much, you know, the numbers are higher. It's just that as a proportion of GDP, it's going up. We're not at the 17, 18% of the US, but that's unsustainable. In West Australia, where I was before, it went from 4 billion to 8.9 billion in a matter of about 6-7 years. That's utterly unaffordable. So government is absolutely interested in minimizing its costs. And I'm going to make a statement here which is going to trouble some because this is what you're up against. I was at a meeting where someone got up and said, we've got to invest in molecular medicine because it will save money. And we said, oh, how's it going to save money? Well, we're going to use a $5,000 test to work out whether a patient who is at the end of their life is going to need a $100,000 drug. How's that going to reduce that down? You're not solving the problem of government. In fact, you're creating a problem. Their problem is that they can't afford enough nurses on the wards to cover shifts. So they said, we don't need handover anymore. Let's stop handover because we don't really need that. Because that saves FTE. So if you're trying to convince and do work in an area where you're saying translational research is important, you've got to pitch the message to a receptive audience. When you've got that going on, they're not that receptive. They're worrying about things like bed days. So if you read the Productivity Commission and you read their research paper on this, which is available on the web and very important and very worthwhile reading, what are the things that they care about? They care about workforce, how they're paying, preventative health. If you can find a strategy to say that we can prevent people getting into hospital in the first place, if you can reduce acuity, you're going to have them as your best friend. They will put money towards those kinds of projects. You can mount an argument. So what is it that you're doing in basic science that can actually prevent a patient fronting up in ED with an acute episode of whatever condition they have? Can you manage them as an outpatient? Can you manage them in a more chronic setting? What other ways can you do care at home? Are there shared care? What can we do with the primary healthcare networks, or whatever they call themselves these days? So the other kinds of things is information and transparency. And I've got, having worked public and private, there's a lie. And the lie is that there's anything like a functioning electronic health record anywhere in this country. It's just rubbish. I did 2 DRGs last year, disease-related group searches on knee replacement and on colorectal surgery. And I asked the question, what is the comorbidities that predict a poor outcome for those patients that are operated on? Is it the surgeon? You know, a lot of people say, oh, well, if the surgeon's fantastic, then you're going to get better outcomes, and it's all down to the surgery. Is it due to the procedure? Not at all. It's entirely about the complexity of the patient when they presented to the hospital. So I asked the question, do we know what the complexity is? Diabetes, cardiovascular disease, BMI, all of these various things, lung disease. Do we record any of that accurately? Do we actually look? What kind of notes do we write up? It's poor, really poor. We couldn't actually answer the question because the data isn't there. And when you go to the medical record and look at the squiggle that's written in there, you know, there isn't an algorithm that translates a squiggle. So the IPA this year put out a tender, in full disclosure I applied for it, and I don't know whether we got it yet, was to create an app to be able to get clinicians to have a drop-down menu to be able to accurately record the data on what they so complications, infections, all sorts of things, so that the coders, the very specialized people who are amazing translators in the hospital, that actually code, which is how hospitals get funded through activity-based funding, can do it accurately and make sure that their activity-based funding and the hospital pricing is efficient. Do you think you can get that information? No. The curious part was that IPA wanted a phone app, and we were sort of pointing out to them that, in fact, more than half of the people that they would require to use this were not permitted to have a mobile phone in the area in which you'd have to fill the data in. So again, translational research, just explaining to someone that you can't use technology in every little area. But there are a whole series of things in here that they want to know about. And Douglas Adams, wonderful author, wrote one time that he loved deadlines. He loved the whooshing noise they make as they sail past. And you can read the Productivity Commission document here and see that. These are all things that it said, 2015, immediate, within a year, immediate. I don't know how many of those have been delivered on right now, but I guess who's counting? So a quality and safety dimension to pricing. This is what troubles government. So what are you doing in basic science that will excite and present to people who are controlling the billions in healthcare to answer their questions? So you can say, well, we're agnostic to that, but this is actually a report which I'm sure, has anyone read this in this room? It's bedtime reading. But this is what the hospitals want to know. They want to know variation in healthcare. So if you're treated in a hospital here, is it different to the treatment that you might receive elsewhere? But you've got to be able to identify it. You've got to actually know why it's there, and then you've got to do something about it. That's really the punchline there. And this is the idea of a self-improving healthcare system. This is a term used by the former Labour government. It's been adopted by the Liberal government, and it is actually part of the MRFF. This is a slide I borrowed from a good friend. Steve Webb is an intensive care physician, and this is the basis of ACTA, the Australian Clinical Trials Alliance, which I'm going to shamelessly plug right now. We've got our Australian Clinical Trial of the Year Award on Friday. There are still some seats available at the Royal Prince Alfred if you want to come along and hear Minister Hunt make the announcement, and Kelso will be there too, and Research Australia and a whole variety. There might be some exciting announcements made at it. But the concept here is that you need clinical registries to be able to understand what you're doing and what's actually going on in a hospital. Then you need to be able to hypothesize what are the things that you need to fix. So you need to talk to clinicians about that. What are the problems that they need to fix? To have a capacity to actually bring your innovation within a clinical trial and a framework about delivering that, and then to implement it. And we haven't got time to go into implementation. Tim's here, he can talk more to that. But that is actually a key. If you do a research study and you have not discussed with the people that are intending to apply it in practice, it's going to be doomed to failure, unfortunately. So what did the Medical Research Future Fund strategy say? And again, I hope you've all read this, those of you who are interested in getting funding. This was the basis for what it was looking at. And the challenges it identified was, we've got a problem with facilitating translation into research outcomes. It should be embedded across the health system, all of these kinds of things, and a lot of motherhood statements. So that's great, and these are all the organisations that are going to deliver on this. Now, one of the problems with this is, can you see specifically where the basic innovation is plugging in? It's not so obvious. And if you look at the strategic platforms here, strategic and international horizons, data and infrastructure, that's about interrogation of clinical data sets and understanding health services and systems, capacity and collaboration, fairly broad, trials and translation, and ultimately commercialization, because they recognize the problem that we have in terms of commercializing our discoveries. But the problem about this is that the teaching, training and research resources are block funded. So I asked the state government in WA, how much do you put into research? And they said, oh, we put in 250 million. I said, oh, it's curious, because no one can actually find that 250 million. Where is it? They ascribe a proportion of every person who works in healthcare and say, you do teaching and training. So they just say 2 hours off every clinician in the healthcare system. So multiply that by all the nurses and doctors. That's how they figure it. So that's not exactly how we should make it work. So this is really my last slide, is saying if you want the world-class research, we actually have to have a focus of how it's the benefits are there for patients, staff recruitment, retention, health outcomes. If you're articulating your message in an environment that is actually difficult now financially, the MRFF is not going to be your saviour. I'm going to put that message out clearly. If you look at the budget papers and you look at where the money's gone, it has directly gone into frontline research. It is going into clinical trials. It is going into those areas, mental health. But how do you then align and ask the question? And the question, and you started to talk about it, Alex, is that we're really talking about what are the questions that you're answering for the clinicians. The clinicians, and it's not necessarily the basic scientist who's now gone into the research, but the problem is we can't afford in medicine at the moment the $100,000 treatment. Everyone wants to be able to do it, but it's going to bankrupt the healthcare system. So if you can provide those solutions through your work and find out what's troubling the clinicians, and you only have to ask them. I sit on the hospital executive, so I ask, what are the things that are troubling you? And it will be things like the 4-hour rule in ED, winter beds, stays in surgery, outbreaks of infection. These are the things that trouble. What are you doing to solve the problem of those people? I'll leave it there because we're going to have a discussion around that. And this is a bit of a challenge to you because it is a difficult one to bridge across. But it doesn't mean that you can't and that it's impossible. Anyway.
I think we'll have a question from Susanna Rafati, who's chair of breast cancer research at Westmead. You're the Thanks, Anna.
Thank you. So, okay, so I'm going to, my talk will be slightly different from Nick's. It's also always, I'm going to actually give you a bit more of a case study, looking just at one sector of our research within our TCRC to give you some examples, if you like, of how we have implemented translational research. Not to say that's the only way that it can be done, but just to give you a little bit of a walkthrough of where we've come from and where we've landed in terms of translational research. So my talk is going to be mainly around ovarian cancer as our case study, and it's very much around basic research embedded in multidisciplinary care. So what does that mean? So we, the way that we do our research at Westmead, we do very basic, a range of research from very basic through to translational in the Westmead Institute for Cancer, for Medical Research as part of the University of Sydney. But we work very intimately with the clinical department, and that has now been formalised through the Sydney West Translational Cancer Research Centre. So we, as you'll see, did a bit of an experiment in terms of actually making research a discipline within multidisciplinary care. I'm also going to, we do a lot of collaborative studies across the country, across the world and across the state. And I'm just going to mention 3 of those studies during this talk. One of them that I will draw on will be the Australian Ovarian Cancer Study, or AOCS. So that was a study that we did across the country. Nick was part of that. In Perth, we had patients from here at from this campus at North Shore. So every major gynae centre across the country essentially was involved in the Australian Ovarian Cancer Study. It was an important study because not only did we recruit large numbers of patients and controls, so well over 3,000 women were recruited to the study. We collected, we've already heard about, biospecimens. We collected high quality biospecimens, tumours and blood, frozen tissue, collected very detailed epidemiological data, but matched to that was extremely detailed clinical follow-up data. So now we're sort of well past 10-year median follow-up on these patients, and we've all talked about the importance of having that rich clinical data. So here was a facility that had all of those elements. And we've actually enabled, because it's we've made it a resource that other people can apply to, it's actually supported well over 100 projects nationally and internationally. And I'm going to tell you just really one of those stories, but it's led to over 200 papers. So we'll talk about AOCS a little bit later, so one of the studies that's come from that. We've also been involved in international studies, here the International Cancer Genome Consortium, so really quite basic. And at the end, I'll just mention Innovate that we've already spoken about, which is a state project. So we do research, very basic research, laboratory research, clinical research, and also involved in multi-centre studies. So as I said, in terms of translational cancer research, it was a bit of an experiment when we started, and it was an experiment that Paul Harnett instituted many years ago. So Paul Harnett is the head of cancer services, and he had this idea that really to do good translational research, we really needed to embed research in everyday clinical care. And so our hypothesis was that, or his hypothesis was, embedding basic research in clinical care would lead to translation of significant clinical problems into research questions. So Connie already made this point, that it's not just one way. It's not just going from the bench to the bedside, but in fact, we need to take the questions from the clinic into the lab, but then also rapidly translate those research results into improved clinical care. Our background, this was the background when we started, that translational research was really the valley of death for basic researchers. So you've got your researchers and your clinician very tentatively on this rickety bridge of one false step, and that could be the end of your career. And so really, that was the background, but what our aim was to build strong bridges and to not have that sort of valley of death, if you like, for basic researchers, that it was okay to dip your toe into a bit of translational research. One of the method that we used in this experiment was firstly to recruit participants. And really, for really good translational research, you need willing researchers, so researchers who are willing to try to modify what they're doing sometimes and to sort of make their research more clinically relevant. But you also have to have willing clinicians. You have to have clinicians who are willing to help researchers to develop their ideas and to recruit patients, etc. So you really have to have willingness on both sides. And also the way that we've set up our research, it's not two pillars. We don't have the clinical group and the research group. We're all part of the same research group. And it's not just one or two meetings. So it's not, the researchers have a lab meeting once a month and invite the clinicians to come along, but actually it's the time that the clinic's on and they usually don't turn up. It's not that sort of, we've really taken the researchers into the clinical domain, and I'll talk a bit more about that later. But really, this is just to show that there's really many points of intersection. It's not just one or two meetings that people attend. There's biobanking that we've talked about that spans it, tumour marker testing, looking at that Connie's already mentioned. PhD students really important. So we've had a number of clinical PhD students who've come into the lab. And over the years, we've sort of learnt to develop research projects for the PhD students where they still have a little bit of clinical work in there, so they feel a bit comfortable, but really challenging them with some very basic aspects of the research. So it's taken a while to get that quite right, but we've but I think that's really that has been very important, is having medical PhD students who come into the lab, and of course things like clinical trials and genetic testing, which we'll talk about. So just to concentrate for a minute on these top aspects, if you like, of the collaboration between the lab and the clinic. One has been the MDT meeting, which I'm sure you all know about. So it's a very clinical meeting, a meeting that's attended by all of the disciplines that look after patients. In our area is gynae cancer, so we have surgeons, pathologists, medical oncologists, but at our centre for about the last 20 years, research staff have been one of those disciplines that attend that meeting. And it's not just me, we have 3 or 4, 5 or 6 people from the lab who attend that meeting. And that has really been incredibly important. And we're very grateful to the clinicians that they feel they've been very welcoming in terms of helping the researchers attend those meetings. The reason that this is really important is that this is the meeting that every case, in our case, is discussed in terms of the pathology, what the surgeons found, what the ongoing clinical care is. But we can come away, I used to keep a book, I can come away from every meeting with 3 or 4 research questions, because it's at that meeting that you really understand what difficulties the clinicians are faced with. When it's the same problem that comes up week after week, you think, oh, you know what, maybe we should do some research into that, because that's clearly an area of need. Whereas then there's other times you think, oh, look, we've got methods that we could do X or Y, but in talking to the clinicians later, they say, look, even if you told us that, it won't make any difference. So you could spend the next 10 years researching that, and we will not change what we're doing. So I think it's been really important for us in terms of really understanding what the questions are and what we are able to do to address it. On a practical point of view, it's been really important for our biobanking, because often the clinical information that you glean from those meetings really help in terms of enriching our biobanking and our clinical data collection. And funnily enough, it's gone the other way. Because the biobankers will sit there and at the end of the discussion say, oh, and what stage was that, or what was the ECOG on that, that in fact it's turned out that the clinicians now, their records have ended up being actually more complete, because the researchers are there asking the question. And so rather than wait for the answer, they look at me like this morning, say, oh, Anna's going to ask the stage, it's blah, blah. So it's an interesting how that has developed. But it's not easy. There's a lot of challenges. The time frames of the decisions made at that meeting, they might have 5 or 10 minutes to discuss a particular case, and it goes really quickly. They might have 20 cases to get through in an hour and a half. Whereas researchers, by the time you think of the question, you might write a grant, won't get through this year, okay, we'll put it in next year. Like the time scales that you're thinking of are very different in terms of sitting at that meeting and developing research questions. So we certainly need other meetings, which are these ones, the research meeting and the clinical trials meetings, where those things can be developed in a bit more detail. The other thing is terminology. I think we can't underestimate when clinicians speak quickly because they've got work to get through, how many acronyms they can use, and it's actually, it takes a while for researchers to be able to understand exactly what's going on. And so that is something that there's certainly a lot of work being done in terms of clinicians going into the lab with practitioner fellowships, etc. We've already had these examples. We don't really have very much in terms of training for basic researchers to understand some of the clinical terminology and clinical aspects. And we're developing, we'll get to later, a molecular tumour board, because what's happening now is, in fact, there's some of the results that we are able to get in the lab. We're getting to the point that some of that can be fed back in real time, and so we're actually developing the mechanisms for being able to do that. So that's quite, it's really quite heartening as a researcher to sometimes be able to contribute to the discussions at these meetings. And I have to say, our surgeon who runs it's fantastic. Whenever we have a result to present at those meetings, she'll always put us on first, because what can happen is if the research aspect might get left at the end of the meeting, and by then everyone's packing up and half the people have left. But you can see the value that they're starting to put on this, that they'll put it on first, so that we deal with that. I've got 5 minutes to get that message across, and then get on with the meeting. So I think certainly it's been a learning experience in both directions. So our case study today is ovarian cancer. I know many of you do research into ovarian cancer, so I'm telling you things that you already know, but I thought there'll probably be very many people who don't. So ovarian cancer is often advanced at diagnosis and widespread and already widespread. About 70% will initially respond to treatment, but the development of resistance is quite common. What you can see here is survival is slowly increasing over time, but it really remains very low. Okay, so we've got a challenge in ovarian cancer in terms of treatment. So just by way of the one little bit of results I want to talk to you today about is just a project that we did around BRCA1 and BRCA2 and ovarian cancer risk. So many of you would know that the BRCA1 and 2 genes were actually discovered back in the mid-1990s. I've chosen this particular area just because there's a few points that I wanted to make. One is that translational research, one researcher doesn't have to do everything. Like, you're building on research that has gone on in the past, and you do your bit in the translational spectrum. I think sometimes people get the feeling that they actually have to do everything. So of course we didn't discover these genes. Okay, they were discovered in the mid-1990s. At that time, we thought that about 5 to 10% of ovarian cancer was familial. That was taken up pretty quickly, and there was at the time recommendation for referral to family cancer clinics for genetic testing was based on women having a family history of breast or ovarian cancer. But the actual rate in Australian women wasn't known. Now, why is it important to know this? The reason it's important is that something that Nick brought up about prevention. Okay, so if a family, here's a family history, so if this woman here, for instance, knows that she has a strong family history of breast and ovarian cancer, there are steps that can be taken to prevent cancer in those women. And now there's actually a difference in treatment. So there's really advantages in knowing BRCA1 or 2 status. So I'm taking this bit of research up in about just prior to 2012, when there was a study came out in Canada that suggested that frequency of 5 to 10% was, the frequency was probably a bit higher than that. Now we had the opportunity in the Australian ovarian cancer study that I had mentioned earlier to actually ask that question. Okay, what is the BRCA1 and 2 mutation frequency in the Australian population? So we undertook a study with David Botel and a number of researchers around the country to measure the BRCA mutation frequency, both 1 and 2. Now, at the time, it was actually quite a large undertaking to do that testing in 1,000 women, but it turned out to be a very productive project. What we found was, in fact, similar to the Canadian study, the frequency was a bit higher than we expected, and in fact, in some subtypes, here a subtype called high-grade serous, it was up to 17%. But one of the most important findings was that almost half the mutation carriers had no family history. Okay, and so in fact they would have been missed. They were being missed in the normal screening methods. So, sorry, let's go back. Oh no. So that paper we published in 2012, the screening guidelines were changed in 2013. So rather than only being based on family history, the screening guidelines were changed so that almost all women with ovarian cancer, regardless of family history under 70, could be referred for testing. Now, because in the session we're talking about translation, how did we get something translated so quickly? The reason was that we actually had, as Nick touched upon, we had the people who were going to use these results involved in the project from the get-go. So the people from the family cancer clinics, the clinicians, they were actually involved in the project from the very beginning. So it wasn't just researchers getting a result and then publishing and hoping to God somebody might pick it up. They actually had all the end users, if you like, involved from the beginning, including consumers. So, and in fact, the new guidelines were drafted before the paper was even published. So I think that this was an important example of how you can get something translated, some very basic research translated quite quickly, and it's to have all of the people involved from the get-go. So, in terms of, so that has turned out to be practice changing internationally. Okay, and so the guidelines have changed here, they've changed internationally, but in fact the uptake wasn't equivalent. Like, it did take a while for people to really get clued into this change of guidelines. So it was something that we had to get it out to all of the clinicians who were looking after ovarian cancer patients. So we undertook a study with Tim Shaw in terms of implementation strategy. So looking at spaced education for clinicians to be able to get that message out, you know, what the guidelines have changed and this is what they are. And that was a very interesting project that we were able to do through our TCRC with Tim. That was published in this paper. We also had referral through MDTs. There's now what's called mainstreaming, like actually turning it into something that's just done routinely for women, all women as they come through the clinic. And there's a project that's been proposed by David Botel and Golly Samemi and others called Traceback, which is okay. It's okay now for women who are being diagnosed now. What about all of the ones that we've missed that are still in the system? So Traceback is to go back and to do BRCA1/2 mutation testing. But really, one of the biggest effects in terms of implementation was a bit unexpected, and that's the Angelina effect. So I think Angelina Jolie probably did more work than any of us in terms of making people understand the importance of BRCA1 and 2, and now it's the patients coming to us asking for it. So sometimes you just can't quite tell how these things are going to be implemented. So, so that's in terms of the screening, and that is really important for prevention, and it's going to be quite difficult for us to estimate how much of an impact that really has. Like, how many women are we going to prevent getting ovarian cancer because of this implementation? But one thing that might be slightly easier is in terms of treatment, because what has now been found is that these treatments, PARP inhibitors, work better on patients who've got a BRCA1 or 2 mutation. So now it's actually important for the patients themselves to know. It's not just for their family. So this is just showing one of the trials where that have used PARP inhibitors to show that increase in treatment. And so olaparib, so that PARP inhibitor, is now available on the Australian PBS for BRCA1 and 2 carriers. So it's in that whole transition through to implementation, to changing guidelines, to being on the PBS. Interesting, I just wanted to mention here, sometimes when you do basic research, you don't really know how that's going to be used in the future, so you just have to always be on the lookout, because what we're able to do here is use that AOCS data as real-world data. Actually helped with the, they use that data in the submission to the PBS. So a different aspect of our work actually helped to get something onto the PBS that is now helping patients to access new drugs. So, yeah, so you don't always know where your research is going to end up. But that's not the end. Of course, we have to get back to basic research. There are resistance mechanisms that arise with some of these BRCA1 and 2 carriers, and of course there's beyond BRCA. because inherited BRCA1 and 2 mutations are only part of the story. Here is the whole spectrum of high-grade serous ovarian cancer patients. Half of this group of patients, this little part of the pie here is BRCA1, but in fact almost half of the pie have what's called HR deficiency. So it's at least one of the members in that pathway, either a somatic or inherited mutation. So half of high-grade serous have got this HR deficiency and may actually respond well to PARP inhibitors. So this is our data, and over here almost exactly the same percentage found with another study in the States. So now it's becoming even more important, not only to measure BRCA1 and 2, but to measure all of those members of the pathway. So there's panel mutation testing, but in fact we're now working on an assay, HRD assay, with, that will actually predict an HRD defect despite what the underlying mutation is. And we're trying to implement that to predict response not only to platinum but also to PARP inhibitors. And we've got a project that Debbie Marsh is part of, which is called Innovate, which I know this is a very busy slide, but just to say that that HRD assay and measuring response to treatment, to get back to Connie's point about having to have biomarkers that will measure response to treatment, is a story that's being repeated through a lot of our research. And so what we've done here is implemented this program called Innovate, and that's to take it's a platform that takes many of those findings that we and others have made over the last decade to actually implement those into everyday clinical practice. And so that HRD assay is just down here. So the idea is that all women, well, the majority of women with ovarian cancer diagnosed in New South Wales will be recruited to the study. We'll be doing a whole, or we are doing a whole spectrum of new molecular pathology tests, so that if they relapse, we'll know what underlying defects are in their tumours and be able to triage them into appropriate clinical trials. And as you can see, there are many people across the state involved in that project work. So we're really hoping that this will be a platform to be able to translate quite a number of our findings. So, just in conclusion, in terms of today's talk and the role of basic science, I think it takes a village to raise a child, but it really takes a village with complementary skills and real commitment to transform scientific discoveries into improvements in patients' care. And also, the example I gave was so that you could see that successful translation research is not always linear, doesn't always go in one direction. In fact, there was a bit of a zigzag, you know, moving from basic to clinical to implementation and now back to basic research again. So it's not always just a linear path. It's also not always possible when you're doing basic research to know where if it will lead to translation or not. I just want to leave you with something I was reading on the weekend. Just in terms of, I think it's really important that there's continued investment in all parts of the translational spectrum. We really can't leave basic research behind. So I've heard from Nick that there's nothing in the MRFF for basic research. NH and MRC funding's going down. I think it's a huge mistake to not fund basic research. This point was being made in this article in Science, where they looked at NIH grants, and they showed that less than 10% of the grants led to a patent. So, and they were saying, well, you know, only 10% of research leads to anything useful. But no, when they looked at the data a bit more carefully, more than 30% of the grants led to a publication that was cited in a patent. So in fact, to get back to that point of everyone plays a part, like it's not every person that will be, every researcher that will be able to span that entire spectrum. That basic research is really very important to fund, as well as all the other parts of the translational spectrum. So I might just, leave it there. It's not my last slide's gone, but where I was going to thank everyone. So thank you. Thank you.
Thank you.
Thank you. So I'm from the Sydney Catalyst and the Centenary Institute. I'm a biochemist, and our fundamental research has led to some small molecules that we've developed that have potential for new cancer therapies. So what I'll do very briefly is just take you through how we've managed to get from T1 to T2, and there's no easy or right way to do this. This is our experience, and I can discuss later in terms of, if we had to do it all again, would we do it any differently? So our research in this space really is about cancer metabolism and tumour metabolism. It's now recognised as one of the hallmarks of cancer. So we now know that tumours metabolize the nutrients that we take into in our diet in a different way than our healthy cells do. And the original observation came from a guy called Otto Warburg, and he realised when he cultured tumour cells in a dish that they had ended up making acid. And from this observation he realised that they were making acid because they were metabolizing the glucose differently. Normally when we metabolize the sugar in our diet, which is our main nutrient, which is the glucose, most of that glucose is metabolized through the citric acid cycle in the mitochondria, and very little ATP is made from glycolysis. But tumour cells tend to switch that a bit, about 50-50 metabolism from glycolysis versus the mitochondria, and that leads to the production of the lactate, which is what Otto observed. And so that's the Warburg effect. In terms of our understanding of it now is that this sort of shift in the metabolism of glucose in particular, it seems to be required for the, the out of control cell division that typifies our tumours. So they find it more efficient to metabolize glucose in this different way because it gets them to the building blocks that enables them to make new cancer cells. So it gets them to that point more efficiently than the sort of metabolism that our healthy cells undergo. Okay? So in general terms, that's our understanding. So because it's really driven by the desire to proliferate, divide as quickly as they can, and to do that, they found that if they metabolize the sugar in this way, they get the building blocks more efficiently to enable them to do that. So the DNA, protein, and lipids. So metabolism is it's probably the most integrated system we have in our cells in our body, and this is a simplified example of how integrated it is. So when we talk about tumour metabolism, this is a very complex system, and interrelated, and all sorts of feedbacks and redundancies built into the system. Our particular focus is mitochondria and a transport protein in mitochondria that we've targeted. So very briefly, our fundamental interest is around this type of chemical modification of proteins that we discovered some time ago, things we've called allosteric disulfide bonds. So a disulfide bond is your covalent bond between pairs of cysteine amino acids in your protein. But traditionally it was thought that these bonds are there to sort of help stabilize the protein and get it through its life. What we've found is that a subset of these bonds are actually functional, in that they're cleaved, and when they're cleaved they can change how the protein works. By analogy, in the same way that some peptide bonds are cleaved in proteins to control how they work, blood coagulation, complement activation, apoptosis, for example, involves cleavage of peptide bonds by proteases. By the same token, there are some disulfide bonds that are cleaved to control protein function. So how we got into this whole business is that we were trying to think of ways to probe this chemical modification in proteins. And what we did is we turned to the periodic table, and we were particularly interested in the metalloids. Now the metalloids are interesting. They have interesting chemistry in that, for example, if we have an allosteric disulfide bond in a protein, it's cleaved. You've got these unpaired cysteine thiols, and some metalloids will cross-link those cysteine thiols, and it's a very tight bond, very tight reaction in living cells. So we thought, okay, let's see if we can make compounds that will do that, because it will enable us to probe the reduced form of allosteric disulfides. We first look at antimony, which is the highest affinity molecule, so that would have been the ideal metalloid to use in this setting. There's very few chemical building blocks using antimony, so we went to the next best one, which is the arsenic atom. And so we made these compounds to probe this chemistry, and one of the ones we made we've called GSAO. So this is an organoarsenical that has a tripeptide backbone. and our arsenical atoms on the end of that phenyl ring there. And so that's the molecule we made. We just basically, we just wanted to make a molecule that was safe, that, you know, the arsenic wouldn't be toxic to the cells. And that turned out to be the case. But when we started using it, we realized it was a metabolism inhibitor. And what it was doing, it was taking out the mitochondria. And look, just very briefly for the fundamental researchers in the audience, what it's targeting is this transport protein known as ANT. This is the molecule, the transport protein that takes the ATP that's made in the mitochondria and shuttles it out into the cytosol, where it's used for all sorts of things. So if you take out ANT, you stop glucose metabolism by the mitochondria. You also perturb mitochondrial function. This leads to proliferation, arrest, and cell death. And the molecule's working in the way that we designed it to do. Is what it's doing, it's cross-linking these two cysteine thiols and ANT that arise when cells start to proliferate. And that's how it's shutting down the mitochondria. So we had some, you know, when we got very fortunate for all sorts of reasons. The molecule was very well tolerated. It's, you know, very water soluble. When we put it in animals, it remained in the circulation. It really didn't dissolve in lipid and get into tissues. And we had some good anti-tumor activity in mice. So then we were keen to see if we could get it into the clinic. And so we looked at various ways of doing this locally. We looked at venture capital locally. We looked at some of the pharmaceutical industries locally. We didn't have really no luck. Then we approached Cancer Research UK. So Cancer Research UK really is a remarkable organisation in the UK that, you know, where they managed to sort of bring together the two biggest cancer charities into an organisation that has great capacity to do big things. And what they will do is do early stage clinical development of interesting molecules that come out of academia. And so we presented this to them and they were keen to take it on. So the deal is that we funded the preclinical work, which wasn't insignificant, cost about $800,000. We had to make the molecule and do the formal preclinical work. And that money mostly came from NHMRC project grants. But they would fund the clinical trial. The clinical trial probably cost about £1.5 million, and that's the element that they funded. So, you know, it enabled us to get into the clinic. So the trial was a phase 1 dose escalation study in patients with solid tumours. So it was an all-comer solid tumour trial. So this is the first patient that was treated. This is at the Christie Hospital in Manchester, and this lady had ovarian cancer. So we, I suppose that the end of the result, we found the molecule was very well tolerated. We defined a dose for phase 2 studies. We had some promising sort of stable disease. The best was 13 months on dosing with GSAO. While this trial was going on, we were interested in trying to make better molecules. And so the thing about GSAO was that, look, let me briefly explain this. The thing about GSAO is a prodrug. It first needs to be converted at the cell surface by an enzyme known as gamma-glutamyl transferase. Then it's taken into the cell, metabolized further before it takes out the mitochondria. So what we thought we'd try to do is just eliminate the prodrug processing, take out the prodrug aspect of it, and we made this compound called PenAO, which is sort of like an end-stage metabolite of GSAO. It's also transport mediated, but it doesn't need any processing. Gets in, takes out ANT. So it had about 10 to 15 times better efficacy than GSAO in animal testing, and it was equally well tolerated. So we thought, you know, this might be a good deal. So we wanted to see, while the GSAO trial was going on, could we sort of progress clinical development of this one? And so we were keen to do it in Australia. Well, look, we did go back to CRUK, but you know, their policy is that, you know, they'll do the first generation molecule, but they won't really look at second generations. They're not really set up to do that. They were interested in doing that. So we were keen to do it in Australia. So we looked at all sorts of options, and then we were lucky to win one of these translational program grants from the Cancer Institute in New South Wales. And that enables us to get into phase 1 studies with PenAO. At the end of the day, that TPG probably funded about half PenAO's development. The rest of the work was funded by some angel investors, some philanthropy, particularly through the UNSW Foundation, also by University of New South Wales, also by New South Innovation. So trying to cobble together all the money to do this phase 1 study about the hardest thing I've ever done. So we've just finished that phase 1 study. This is the first patient treated by Jayesh Desai, whom some of you may know, at the Royal Melbourne Hospital in Melbourne. So it was a 3-centre trial, Lighthouse here in Sydney with Lisa Horvath, Jayesh at the Royal Melbourne Hospital, and Hamilton at the Peter Mac in Melbourne. So again, it was a phase 1 dose escalation, solid tumours, all comers, and I suppose it's very similar to results to the GSAO trial. The molecule was well tolerated. We got to dosing for phase 2 studies at blood levels that should be efficacious, and there was some sort of stable disease, no partial responses or complete responses, which would have been the ideal scenario. So where we are now is trying to sort of progress the clinical development of PenAO. So at the end of the day, we had to make a call. We think PenAO had better efficacy in animal studies, so we decided to sort of stick our time and effort and our resources into PenAO's development. We'll probably never know if that's the right decision, but that's what we decided to do. And so, you know, when we go into phase 2, we need to combine it with something else. So we're looking at various combinatorial options. And we're trying to sort of do this in a mechanistic way. So if we take out ANT, let's try to inhibit hexokinase 2, which is a component of this process, of this complex. Hexokinase 2 activity in the cytosol is in turn regulated by TORC. There's some good inhibitors of TORC, the rapalogs, and when we combine PenAO with the rapalog, we get very good combinatorial efficacy. And it's really quite toxic for tumours. When we treat established tumours, we see sort of good necrosis. So now we're trying to find the funds to do a phase 2 study with this combination. And that's pretty much where we are. I've spoken to probably every pharmaceutical company that I've ever heard of, and you know, lots of venture capitalists, and the overwhelming message is that, look, we need phase 2 data. We want to see some phase 2 data. If we see something positive in phase 2 data, come back and tell us, and that's fair enough. The other sort of challenge that we have is, you know, you guys would know as well, girls that would know as well, is that, you know, sort of oncology drug development right now it's all about the immune checkpoint inhibitors, because of some of the spectacular success that have been had. So it's very hard to get traction, you know, in this space right now if you're working in tumour metabolism and not tumour immunology. All these sort of factors come into play. Anyhow, so that's where we're at. And there's lots of people working at both in the fundamental level and also the clinical level that have been involved in this process, and I've listed some of them here. So I'll stop there.
Might have